Good afternoon, everyone. My name is Eric Hopkins. Welcome to my second seminar for my PhD program. My research focused on Sideroxylon mascatense. It is a wild fruit-bearing plant you may have tried while visiting the mountains here. In my research, I studied the population genetics and the distribution and associated plant communities of this species. The rugged and steep Hajar Mountains dominate the north of Oman, with peaks of up to 3,000 meters. Oman hosts over 1,400 wild plant species, with 191 of them being range restricted, and 72 are rare and threatened. 703 of the over 1,400 species are found in the mountains of northern Oman. Of the 703, 298 species belonging to 73 families are found above 1,500 meters in the western Hajar Mountains. Of these, 22 are range restricted and 33 are classified as threatened, near threatened, or data deficient. This is a view from Jebel Hat at about 2,000 meters in the western Hajar Mountains. This is a view from an area just west of Jebel Shams at about 2,000 meters in the western Hajar Mountains. Here you can see the sparsity of the high mountain woodlands. The western Hajar Mountains exhibit a variety of seasonal microclimates that are generally more temperate and Mediterranean when compared to those occurring to the rest of Oman. The Sheikh Meteorological Station on Jebel Akhdar has recorded average temperatures that are 12 degrees cooler than those in Muscat. The mountains had an average rainfall over the last 2 decades of 210 millimeters, while the meteorological station in Muscat only recorded an average of 68 millimeters. The data from Sheikh also shows that over the last 20 years, there have been an increase to the minimum temperature of 0.6 of a degree per decade, an increase in mean temperature of 0.1 of a degree per decade, and precipitation of 33 millimeters per decade. This data came from the National Center for Information and Statistics weather data online. In this data set, there were quite a few missing data points, especially for precipitation. Also, data only went back to the year 2000 for the Sheikh weather station. I found 2 studies that reported a similar increase in temperatures, but they both recorded a decrease in precipitation. Al-Kabani et al. Analyzed over 30 years of data from the Sheikh Meteorological Station on Jebel Akhdar and showed an increase of minimum temperature of 0.8 of a degree per decade, an increase in mean temperature of 0.3 of a degree per decade, and a decrease in precipitation of 9.4 millimeters per decade. Similarly, Al-Sarmi and Washington found similar results from 1980 to 2008. Moreover, Ludling et al. 2009 found that between 1983 and 2008, there was an average decrease in chilling hours of 1.2 to 9.5 hours per year. Using this data, they projected into the future with 2 different modeling scenarios. One predicted 13%, and the other 75% of the years would have insufficient chilling hours for pomegranate fruit production, the major tree fruit crop in the mountains. In general, these reports quantify and show the changing climate of the western Hajar Mountains. Temperatures have increased while precipitation has decreased. This really puts pressure on an already stressed plant communities in the western Hajar Mountains. Sideroxylon mascatense is a key species in the higher elevation plant communities found in the western Hajar Mountains of Oman and is associated with junipers, polycarpos, var cerevaschanica, Olea europa, subspecies cuspidata, Dodonea viscosa, and Ziphus hajarensis. This photo shows a cluster of juniper, olive, and Sideroxylon mascatense growing together on Jebel Akhdar at about 2,300 meters. In northern Oman, Sideroxylon mascatense grows throughout the mountains, with the largest populations found at elevations from 1,000 to 2,400 meters. Sideroxylon mascatense is also known to exist in Dhofar. Sideroxylon mascatense is an evergreen plant growing to heights of up to 4 meters that can be classified as a large shrub or a small tree. This is a large specimen of Sideroxylon mascatense on Jebel Akhdar at about 2,300 meters. It is approximately 4 meters tall and consists of multiple trunks, possibly multiple specimens, growing together to form a large cluster. Sideroxylon mascatense can live for many years. This is an example of a very old specimen with quite intricate burls, as you can see. The wood of Sideroxylon mascatense is quite dense. Other members of this genus are harvested for their timber in Central America. Typically, the branches are well armed on Sideroxylon mascatense. This provides some protection from goats and sheep and donkeys that graze and browse on specimens throughout the year. This specimen was quite unique, having a heavily armed main trunk. Flowers develop on new growth that occur from January to April, with fruit maturing from April to September, depending on elevation. On your screen now, in A, you can see the flowers and floral buds, and in B, you can see the newly formed immature fruit of Sideroxylon mascatense. 2019 was an interesting year for fruiting specimens in the mountain. While the weather pattern didn't vary from the 10-year averages, many agricultural crops flowered and fruited earlier than expected. Most of the specimens of Sideroxylon mascatense fruited during the summer as normal. However, there were some that started flowering and fruiting much earlier. This specimen had well-developed unripe fruit in mid-February of 2019. There are two distinct morphotypes of Sideroxylon mascatense in Oman. The fruit of this species can be found when available and collected from the wild in the small local markets or sold along the roadside. Known for its sweet taste, the fruit is consumed fresh when in season, but it is occasionally consumed dry as well. It has also been shown to be a good source of antioxidants. The most common morphotype, known locally as boot, has a small purple-black fruit. The other less prevalent morphotype is called hajimt. Its yellow-brown fruits are more sought after for their flavor. The fruit from this morphotype is known to have a lower total soluble solid content than the purple-black morphotype. Here you can see for a comparison the flowers and fruit of the two morphotypes of Sideroxylon mascatense found in Oman. You can see in A, the flowers from the boot morphotype, B, the flowers from the hajimt morphotype, and C, an example of the boot fruit, where in D is the hajimt fruit. While the fruit is drastically different in color, the flowers have no visible difference, and there are no other morphological differences between the two morphotypes. The size of the fruit varies greatly. The specimen shown here, the purple-black morphotype, had fruit up to 19 millimeters in diameter. It was found on Jebel Akhdar, growing directly in the water flow of a very steep-walled wadi. The seeds of the larger fruiting specimen remained the same size as to what was observed with some of the smaller fruit. The seeds of Sideroxylon mascatense have a hard, dense seed coat, which impedes germination. One propagation study showed mechanical scarification to be the optimal strategy for seed germination. Another study used the fungus Rhizopus stolonifera to digest the seed coat to promote germination. Globally, Sideroxylon mascatense is found from North Africa, the Middle East, to parts of Asia. In Pakistan, only the purple-black morphotype has been reported. This photo is from an article by Ahmed et al. In 2019. In North Africa, only the yellow-brown morphotype has been reported. Very little research has been documented on Sideroxylon mascatense in North Africa. I found it mentioned in the flora of Somalia, and it was also mentioned in a vegetative community study in Ethiopia. The greatest amount of research on Sideroxylon mascatense comes out of Pakistan. There has been one vegetative community study on it specifically, and two others with Sideroxylon mascatense as an associated species. Most of the research from Pakistan focuses on the medicinal properties of Sideroxylon mascatense, whether from the leaves, fruit, or bark. These projects show the incredible antioxidant, anti-arthritic, antimicrobial, anti-tumor, and antibacterial properties, among others, of this underutilized superfruit. All of the existing medicinal research has utilized the purple-black morphotype that is found in Pakistan. To date, no medicinal or physicochemical studies on the yellow-brown morphotype have been conducted. The main objective of my research was to produce a description of the distribution, associated plant communities, and population genetics of Sideroxylon mascatense in the Western Hijra Mountains of Oman. Specifically, to map the distribution of the species in Oman, to describe the species' growth forms, ecological habitats, and associated plant communities, to conduct a molecular characterization of the Omani wild specimens, to determine the mineral content difference between the soil, leaves, and fruit, and to determine the mineral content difference between the two morphotypes. I broke my research project into three main tasks: a botanical survey, a molecular analysis, and a mineral content analysis. As methodology was the focus of my first seminar, I'm not going into it now, as time does not permit. However, I'd be happy to address any questions pertaining to the methodology during the Q&A session at the end. I'll start by discussing the results of the botanical survey. In order to map the full habitat of Sideroxylon mascatense in Oman, herbarium records from the Royal Botanic Garden, Edinburgh, the Oman Botanic Garden, the Oman National Herbarium, and the Sultan Qaboos University College of Science Herbarium were added to records from a previous study, unpublished work, and the quadrat locations from the current study. The bulk of the population distribution in the Western Hijra Mountains between Jabal Akhdar and to the east and Jabal Shams to the west. Small isolated populations exist on Jabal Aswad and are scattered elsewhere throughout the Eastern Hijra Mountains. In Dhofar, several scattered populations have been documented on Jabal Qamar and Jabal Samhan. No specimens are known to exist in the Western Hijra Mountains from Jabal Misht to north to Musandam. This is a distribution map of Sideroxylon mascatense in Oman. The upper section is showing populations in the Eastern and Western Hijra Mountains, and the lower section showing populations in Dhofar. Green dots represent locations where Sideroxylon mascatense either currently exists or was previously reported. The studies took place between June 2018 and August of 2019. Quadrats were surveyed in June, July, and August, as this is the optimal fruiting period for Sideroxylon mascatense in the Western Hijra Mountains. There were 6 quadrat locations that were impossible to physically access, so 3 accessible locations in the appropriate strata were chosen to ensure that a minimum of 17 quadrats were surveyed in each stratum, not including the extra quadrats of the yellow-brown morphotype specimens. Given the infrequency of occurrence of the yellow-brown morphotype, when specimens of this morphotype were located, an extra Swati quadrant was added around them. In order to access the 114 quadrats surveyed in the study, I personally hiked over 200 kilometers over the 2 summers, wearing out 2 pairs of boots in the process. Actual elevations of the 114 quadrats surveyed in this study ranged from in altitude between 1134 and 2,419 meters above sea level. There were some specimens of Sideroxylon mascatense found at elevations as low as 800 meters. However, they are rare and located in protected areas with northern aspects. The altitude limitations seem to be related to water availability more than temperatures. High temperatures were noted throughout the habitat range of Sideroxylon mascatense. Obviously, there could be temperature requirements for fruiting and flowering, as many of the lower elevation specimens, especially on the south side of the mountains, did not fruit during the study period. The health and vigor of the specimens seemed to be more related to water availability. The specimens growing in wadis or in drainage areas were larger, fuller, and had more fresh vegetative growth. If fruiting, specimens in areas with more available water had larger and more abundant fruit as well. The higher elevation locations in the mountains, while maybe receiving more precipitation, the surface water doesn't stick around. It runs off in pools at lower elevation areas. I believe this is why the greatest population densities occurred in the mid-elevation zones, as well as more frequent, heavier fruiting. This is a map of the study area in the Western Hijra Mountains. The populations of Sideroxylon mascatense specimens per quadrat location were classified into 5 size classes. The quadrats with the largest population of Sideroxylon mascatense were found at elevations between 1360 and 2350 meters in the eastern half of the study area on Jabal Akhdar. These are the large green circles you can see in the lower right of the map. Overall, a total of 513 specimens of Sideroxylon mascatense were located. Of these, only 34%, 172, had mature fruit. And of those 172 fruiting specimens, 158, which accounted for 92% of the total fruiting, were the purple-black morphotype, and 8%, or 14 specimen, were the yellow-brown morphotype. Sideroxylon mascatense ranged from 0.5 meter to 5 meters in height, with drip lines from 1/4 meter to clusters of up to 4 meters in diameter. In this map of the study area, the circles locate the quadrats with different colors representing the morphotypes of the fruiting specimens. If no mature fruit were present at the time of the survey, the quadrats are indicated as no fruit. The purple-black morphotype was present in all but 1 quadrat, where the yellow-brown morphotype was observed. Throughout the survey, 54 perennial non-cultivated species from 32 families were observed growing with Sideroxylon mascatense. The three most species-abundant families were Asteraceae with 14% representation, and Fabaceae and Lamiaceae, both with 11% representation. Of the species associated with Sideroxylon mascatense, 8 are regionally endemic, 3 are endemic to Oman, and 2 are considered rare and threatened. The 5 most common species found were Dodonea viscosa, Olea europa, subspecies cuspidata, Euryapis arabicus, Sagarethia thea, and Grewia erythraea, which were found in 80%, 63%, 56%, 51%, and 48% of the quadrats. Average species richness across all quadrats of the study was 7.4. The average species richness had a weak yet significant negative correlation with elevation, an R value of -0.27 with a p-value of 0.003. Shannon's diversity index followed a similar trend with an average of 1.2 and a weak yet significant negative correlation with elevation, an R value of -0.23 with a p-value of 0.013. This photograph was taken of Sideroxylon mascatense growing in a moderately lush wadi at the mid-elevation of its habitat range. The first division of two-way cluster analysis was utilized for the community study. This division produced two groups accounting for 87.5% of the variation in the data. With these two groups, the indicator species analysis gave the most significant indicator species with the lowest average significance level. These two groups are the two main plant communities associated with Sideroxylon mascatense and fall along an altitudinal gradient. The communities were named after the two species with the highest indicator value in the group. These were the Acritocarpus orientalis Euphorbia lyrica community and the Olea europa Euryapis arabicus community. Twin span analysis corroborated the two-way cluster analysis grouping. Using the groups indicated by two-way cluster analysis, the multi-response permeation procedure showed a highly significant difference between the groups. Using the main matrix of presence-absence data, it gave a within-group agreement A of 0.2, a test statistic T of 53.5, and a highly significant p-value. When used in the matrix of environmental variables, the within-group agreement A was 0.43, and the test statistic T was 55.7 with a highly significant p-value. Non-metric multidimensional scaling ordination plotted the sample units in species space along an altitudinal gradient. A two-dimensional solution was chosen for the final solution because the mean stress in the 250-run Monte Carlo test was 16.9 at a p-value of 0.012. The best solution had a final stress of 17.1 after 55 iterations. Two-way cluster analysis groupings of the sample units, the colored triangles here, are overlaid with Group 1, the lower elevation community, the Acritocarpus orientalis Euphorbia lyrica community, outlined in red, and the Group 2, the higher elevation community, the Olea europa Euryapis arabicus community, outlined in green. Species are represented by the blue dots. These are the 8 significant indicator species with the highest indicator values for the lower elevation community associated with Sideroxylon mascatense. Quadrats aligned with the Acritocarpus orientalis Euphorbia lyrica community were found at elevations from 1134 to 1925 meters. This was the most diverse community associated with Sideroxylon mascatense, with an average species richness of 8.7 and a Shannon's diversity index of 2.1. It also had the most diverse quadrat in the surveyed in the study, with a species richness of 17 and a Shannon's diversity index of 2.8. The Acritocarpus orientalis Euphorbia lyrica community describes the lower Sideroxylon mascatense habitat range, with an average elevation of 1,345 meters. The habitat ranges from steep shale-covered hillsides, rolling hills and summits, as well as wadis. The soil structure of this community varies from very rocky in places to rocky clay silt packed in runnels between exposed rock slabs. Quadrats surveyed at the higher elevation of the limits of this community were located in steep hillsides with little access to groundwater. The top 5 species of this community are Grewia erythraea, Acritocarpus orientalis, Euphorbia lyrica, Polygala mascatensis, and Vachelia Gerardii subspecies negavensis, which occurred in 83%, 79%, 79%, 69%, and 62% of the sample plots. This photograph shows an example of the lower elevation community. It was taken at about 1,200 meters above Wadi Mistal. These are the 8 significant indicator species with the highest indicator values for the higher elevation community associated with Sideroxylon mascatense. Quadrats aligning with the Olea Europa Euryoptes arabicus community were found at elevations between 1,320 and 2,419 meters. The average species richness for this community was 6.96, with a Shan's diversity index of 1.9. The Olea Europa Euryoptes arabicus community, with an average elevation of 2,067 meters, associates with Sideroxylon mascatense at the higher elevation limits of its habitat. The habitat ranges from gently rolling hills, hillsides with moderate to steep slopes, to areas nearer in wadis. Many of the quadrats of the lower elevation limits of this community were located in wadis or on steep, protected north-facing hillsides. The top 5 species according to the relative frequency in this community are Dodenea viscosa, Olea Europa subspecies cuspidata, Euryoptes arabicus, Sigarithi athea, and Tacuria mascatense, which has occurred in 96%, 84%, 75%, 62%, and 51% of the sample plots. This photo shown here an example of the higher elevation community. It was taken from about 2,200 meters between Jebel Shams and Jebel Sara. The results of the botanical survey and plant community analysis aligned with previous vegetation surveys in the Western Hajar Mountains. This is a map of the quadrats from the 2 communities in the current study, overlaid on the model created by Brinkman et al. In 2011. My study encompassed an area much larger than the one modeled here, and therefore only 2 quadrats from the lower elevation community fell within the model's extent. Within the model's extent, the quadrats belonging to the Olea Europa Euryoptes arabicus community fell within the upper 2 communities of the model 91% of the time, and the other 9% falling in the lower elevation communities of the model. Insect, bird, and animal associations with Sideroxylon mascatense were not a direct part of my study. However, these associations point to the importance of this species as a viable member of its ecosystem. Therefore, I noted any interactions with insect, bird, or animal specimens observed with Sideroxylon mascatense and associated plant communities. Goats, sheep, and donkeys were observed and are known to graze and browse, sometimes quite heavily, on Sideroxylon mascatense and associated species. Birds find safe harbor and nest in the branches of Sideroxylon mascatense, as you can see in the picture here. Fox species have been known to feed on the fruit. This is feces from either a Blandford's fox, Vulpes cana, or red fox, Vulpes arabica, found on Jebel Shams with Sideroxylon mascatense seeds present and a purple hue from the fruit of the purple-black morphotype. Here we have a bagworm larva in its cocoon on Sideroxylon mascatense and a cicada on a branch of Sideroxylon mascatense. In the photo here, you can see A, leaf miners on Sideroxylon mascatense leaves, and B, the adult Eristalis tenax, known as the rat-tailed maggot-fly, is a visitor and possible pollinator of Sideroxylon mascatense flower. I'll now move on to the weather-related data that I recorded during my study. The weather in Western Hajar Mountains during the period in which the surveys were conducted was similar to the trends set in the past 30 years. The average rainfall recorded on the Sake Weather Station on Jebel Akhdar was 156 millimeters in 2018, 325 millimeters in 2019, compared to the 221 millimeter 10-year average between 2010 and 2019. According to the data loggers placed in the fields, the months with the highest mean temperatures were June at 25.5 degrees, July at 25.1 degrees, and 24.3 degrees in August of 2018. The hottest temperatures were recorded in August 2018 at 45.7 degrees on Jebel Hat at about 2,300 meters above sea level. The coolest months were December 2018 with a mean temperature of ½ degree Celsius, and January 2019, which had a mean temperature of -2.2 degrees Celsius. The highest mean relative humidity occurred in November 2018 with 67%, December 2018 had 63%, and 54% in January 2019. All months had occurrences of maximum relative humidity between 90 and 100%, except for June 2018, which peaked at 84%. Moving on to the molecular analysis. This is the map of the locations from where the molecular samples were collected. The quadrants were selected at random. However, since fruit color is the only morphological trait separating the morphotypes, only samples from the fruiting specimens were utilized in the study. This excluded some of the originally selected quadrats, as the specimens located there were not fruiting. A total of 35 specimens of the purple-black morphotype were utilized in addition to 10 specimens of the yellow-brown morphotype. They were analyzed by AFLP fingerprinting. Out of the 10 different primer combinations, 5 were analyzed for the population study. Of these 5 primer combinations, 2 gave consistent results and were utilized in the study. These were the primer combinations number 2 and number 5 in this table. The analysis of Sideroxylon mascatense populations for genotypic and gene diversity was performed using 2 primer pair combinations that produced 260 polymorphic bands. The analysis revealed low to moderate levels of nice gene diversity for Sideroxylon mascatense. Genetic diversity of the purple-black morphotype at 0.2131 was higher than that of the yellow-brown morphotype at 0.1751. Each individual specimen was found to be a separate genotype, giving a total of 45 genotypes for the 45 specimens sampled. The 2 morphotypes showed a moderate to high level of genetic similarity based on Nye's unbiased measure of genetic identity. Analysis showed that individuals ranged from 69.8% to 96.9%, with a mean of 87.7%. The purple-black morphotype showed the broadest within population similarity range at 69.8% to 95.7%, with a mean of 85.7%. Overall similarity between the populations of the purple-black and the population of the yellow-brown morphotype was 97%. Overall genetic distance between the populations of the purple-black morphotype and the population of the yellow-brown morphotype was 3.04%. This is an unweighted pair group with arithmetic mean dendrogram illustrating the genetic differences for 45 samples of Sideroxylon mascatense and 2 outliers based on AFLP fingerprinting using 299 loci. Cluster analysis separated the specimens studied into 2 main clades, one having majority purple-black morphotype and the other having a majority yellow-brown morphotype. Samples are identified with a B or an H representing boot or hand. 2 outrooting samples, lime and sapodilla, clustered individual clades, distinct from the 2 Sideroxylon mascatense clades. Sapodilla is a member of the same family Sapotaceae as Sideroxylon mascatense. A moderate and significant level of genetic differentiation was found among the 2 morphotypes of Sideroxylon mascatense, with a fixation index of 0.1037 at a highly significant p-value, with a number of migrants at 2.6, according to the analysis of molecular variation. Analysis showed 10.4% variation among the morphotypes and 89.6% variation within. Pairwise genetic differentiation among the morphotypes also showed a moderate and significant level with a fixation index of 0.1037 at a highly significant p-value. The number of average pairwise differences within the populations of the purple-black morphotype was considerably higher at 61, where the yellow-brown morphotype had 6.7. The corrected average pairwise difference among the 2 morphotypes was 50.3 at a highly significant p-value. Now moving on to the mineral content analysis. Mineral content was not a large part of my study. However, the work I did lays a good foundation for much needed future research in this area. The samples were analyzed for macronutrients required for plant growth, as well as the micronutrients and heavy metals identified by ICP. Of these elements, only nitrogen, phosphorus, potassium, calcium, iron, magnesium, sodium, and zinc were found in the samples. Manganese was found in the soil but not in the plant samples. The electroconductivity of the soil samples as well as the pH were also recorded. No significant differences were found at any level for EC or pH. Average EC across all the samples was 1,652 microsiemens per centimeter compared to the control of distilled water at 736 microsiemens per centimeter. Average pH across all the samples was 8.5 compared to the control of distilled water at 8.6. The mineral content of the soil was analyzed by 4 different grouping variables: morphotype, stratum, site of mountain range, and elevation class. Nitrogen and sodium levels were the only elements that showed significant differences. The mineral content of the leaf samples were analyzed by the same 4 grouping variables. Nitrogen was the only element that showed any significant differences here. The mineral content of the fruit was analyzed by the same grouping variables as well. Nitrogen and magnesium were the only elements that showed significant differences. However, the levels of essential minerals that are known as antioxidants were quite high compared to other fruits. A quick comparison of the concentrations of calcium, magnesium, and zinc found in Sideroxylon mascatense fruit showed that they were considerably higher than those found in other fruits. Having summarized the mineral content analysis, I now point your attention to this specific finding. There was a significant difference in the levels of magnesium between the 2 morphotypes studied. The yellow-brown morphotype had a significantly higher mean at 1.6 grams per kilogram than the purple-black morphotype at 1.46 with a p-value of 0.044. This shows that future research needs to focus on comparing the nutritional and physical-chemical character attributes of both morphotypes of Sideroxylon mascatense to known varieties of superfruits. As I mentioned earlier, all existing research on the medicinal attributes of Sideroxylon mascatense have been performed on the purple-black morphotype. This study is the first to identify the associated plant communities of Sideroxylon mascatense in the Western Hajar Mountains, and the first to define its distribution throughout Oman. As such, with the largest and most productive wild populations identified, the optimal elevation and habitat range have been identified. The results from the study can now be used to promote the conservation of Sideroxylon mascatense in this region, as well as identifying the optimal location for development of Sideroxylon mascatense as a cultivated agricultural crop. This is also the first study to analyze molecular characteristics of Sideroxylon mascatense, in which a distinct separation was found between the morphotypes. The results have provided a base for future studies utilizing next-generation molecular techniques to isolate the genetic differences responsible for the separation of these 2 morphotypes in order to develop breeding programs that can produce reliable cultivars for commercial agricultural purposes. This study was also the first to analyze the mineral content of the yellow-brown morphotype and compare it to the purple-black morphotype. This also lays important groundwork for the future research comparing the nutritional and physical-chemical attributes of these 2 morphotypes with those of known superfruits. Recommendations for future research would be to utilize next-generation molecular tools to identify genes that are responsible for the difference in morphological characteristics such as morphotype, fruit size, and nutritional and medicinal qualities. This information would be beneficial in breeding programs aimed at the domestication and cultivation of this underutilized plant species. Research should also focus on the propagation and cultivation techniques to allow for replanting in areas damaged by human activities, as well as for commercial agricultural projects. Studies focusing on the nutritional and physical-chemical attributes of both morphotypes in comparison to known superfruits should also be conducted. This would help establish Sideroxylon mascatense as a superfruit in the global and local markets. I would also recommend studies on the associations of the entire ecosystem that Sideroxylon mascatense is part of in the Western Hajar Mountains. Research into the habitat, phenology, and associated species of Sideroxylon mascatense in the cloud forests of Dhofar is also recommended, as the seasonal monsoon climate changes the seasonality and could change the attributes of this underutilized fruit-bearing wild plant in Oman. Throughout my study, I found no recruitment of Sideroxylon mascatense in the entire study area. None in the quadrats themselves, and none while trekking over 200 kilometers to and from the quadrats. Therefore, no recruitment of Sideroxylon mascatense was observed, which could lead to diminishing populations. This could be due to climate change increasing the mortality of the seedlings, or because of the overgrazing by domestic and feral animals. This, along with dying mature specimens at the extremes of the habitat range, will lead to diminishing populations. As this study has shown, Sideroxylon mascatense is associated with several endemic and threatened species in Oman. Protective measures therefore need to be taken to preserve the fragile ecosystem of the Western Hajar Mountains and ensure the future survival of Sideroxylon mascatense and its associated communities. Thank you very much for your time and attention.